Hello and welcome back to the MCAT grind. The light is a little bright, but I'm gonna make do. So, we have Biochem Chapter 7, RNA and the genetic code. Now it's a quick review, because this should be pretty rudimentary. DNA, RNA to protein is the central dogma of biology. Of course, we have the coding versus the template strand, which the coding plate is not directly binded by the RNA polymerase because you bind to the template, and because it's complementary, you actually code out what the coding strand entails. Now, the different RNA polymerases, 1, 2, 3, all code for different type of RNA. rRNA, which is ribosome RNA, is being used by RNA polymerase 1, and RNA polymerase 2 synthesizes mRNA, or heteronuclear RNA, which is the precursor RNA before it matures to mRNA. Right, and RNA polymerase 3 synthesizes tRNA, which is responsible for bringing the amino acids over to the growing chain of protein during translation. In transcription, we have a similar start of helicase and topoisomerase unwinding the DNA. After unwinding, instead of having a primer set down, transcription doesn't need it, and it just uses RNA polymerase 2 to bind to the promoter region directly, and the promoter region has interesting point about 25 base pairs upstream called Tata box because it's high thymine and adenine content. Now once it's binded to, then it can start synthesizing a strand which is complementary to the template strand, and it's pretty much the same as the coding strand, in exception that uracil replaces T. Once the heteronuclease RNA is made initially in the nucleus, it then quickly splices out the introns and joins together exons. Depending on the type of protein product being made, there could be alternate splicing. Maybe perhaps some exons are actually removed out as well in order to make a different type of protein. Now we have some modifications to add. Perhaps you can join a 5' cap and a poly A tail, and 5' cap just keeps it from being degraded, and poly A tail similarly. But poly A tail also, with the longer chain it is, the longer the lifespan of the protein. So if it's only going to intracellular space, it might not even need to go grow a long poly A tail. Next we have translation. Translation has initiation, elongation, and termination. Initiation happens at the Chine del Garno site, and then we have elongation following the attachment and the protein the peptide growing chain, and the exit spot, where first the tRNA is brought to the active spot, and then the protein is transferred onto the growing chain, and the tRNA exits and can go pick up another amino acid. Termination is done by hydrolase, which adds a water molecule there and to cut off the end and stops the chain. This is in conjunction with the protein or with the amino acid coded with the UAA, UGA, or UAG. Those don't actually code for an amino acid, but those are the terminations. So you can remember them, the abbreviations by you are annoying, you go away, you are gone. In the post modifications, we could do the following. Either chaperones could help us fold the protein better. We could phosphorylate the protein to activate or inactivate it, such as in phosphate kinase and such. We also have carboxylation, which allows an attachment site from calcium. Glycosylation could be done in the Golgi apparatus. And we also have prenylation. Prenylation just adds different lipid molecules to active enzymes. Now finally we have operons. Operons are basically products of whether they can be controlled better in the regulation of protein production. And we have two main types of operon to study: inducible system and repressible systems. These are all the basic structures of operons. starting from the structure component, which codes for the actual protein, the operator spot, which allows repressible protein, the repressor, to attach, the promoter, in which the polymerase, RNA polymerase 2 attaches, and we have the regulator, which produces the repressor protein. What happens is the regulator regularly produces the repressor protein, which then binds to the operator, and that inhibits the promoter binding RNA polymerase to move past it, right? Because it's physically stuck there as a repressor, and the RNA polymerase chain can't get past it to interact with their structure component. However, in an inducible system, there could be certain molecules that come in and bind to the repressor and remove them from attaching the operator. Now this frees up a spot for the promoter to keep going, and then results in increase in protein product production. The repressible system, however, has the regular structure constantly being made because there's no good repressor actually binding onto the operator protein. It's just half part of a repressor. So this is already very active. However, in this system, the repressible system, you could have a cofactor that comes and bind to the repressor and make it fully functional and allows it to bind to the operator, which now then blocks the promoter from going on to translate. In an inducible system, we have the lac operon, and in repressible system, we have the trip operon. So inducible system where if lactose is present in the medium substrate, then lactose could then bind here to the promoter region and enable the regulator or enable the production of certain factors that lactose bind to the repressor and knock it off of the operator. In a repressible system, tryptophan is regularly being produced constantly, but the availability of tryptophan in the environment binds to the repressor, and then they co-repress and bind to the operator, and thus cease the production of its own tryptophan. Alright, and that's pretty much it. Take care.